Hey there, I'm your name, and welcome to a deep dive into the world of polynucleotide chains. These chains, as you might guess from the name, are made up of lots of nucleotides linked together. And as we all learned in high school biology, those chains make up DNA and RNA, the blueprints of life itself. Today we'll break down the topic into easy-to-understand steps, from the basic building blocks of nucleotides to the intricate double helix structure of DNA. We'll explore how these chains are formed, understand their unique properties, and see how their structure dictates their function in the grand scheme of genetics. So whether you're just starting your biology journey or need a refresher on these fundamental concepts, stick around. This video is your one-stop shop for understanding the fascinating world of polynucleotide chains. Before we dive into the complex structure of polynucleotide chains, let's start with the basics. The nucleotides. Imagine these nucleotides as individual Lego blocks. Just like Legos, they're the building blocks of something much bigger. Each nucleotide has three main parts: a nitrogenous base, a 5-carbon sugar, and a phosphate group. Now, the nitrogenous base is the exciting part. There are 4 types: adenine, A, thymine, T, guanine, G, and cytosine, C. In RNA, uracil, U, takes the place of thymine. These bases are like the code letters of our genetic alphabet. The 5-carbon sugar, either deoxyribose in DNA or ribose in RNA, acts as the backbone of the nucleotide. Finally, we have the phosphate group, which plays a crucial role in linking these nucleotides together. These 3 components together, the base, the sugar, and the phosphate, form the fundamental unit of a polynucleotide chain. Now that we've met our nucleotides, let's see how they come together to form chains. Remember those phosphate groups we talked about? They play a crucial role in linking the nucleotides together through a process called a phosphodiester bond. Think of it like this. Each nucleotide has a head, the phosphate group, and a tail, the sugar. The phosphate group of one nucleotide likes to hold hands with the sugar of another nucleotide, creating a strong bond between them. This hand-holding, or phosphodiester bond, continues down the line, creating a long chain of nucleotides, a polynucleotide chain. But there's another crucial connection happening here. Remember those nitrogenous bases? They're not just hanging around, they're looking for a dance partner. Adenine, A, always wants to pair with thymine, T, or uracil, U, in RNA, and guanine, G, always pairs with cytosine, C. They do this through hydrogen bonds, forming the iconic base pairs that hold the 2 strands of DNA together. So far, we've built a chain of nucleotides and linked them with their base pair partners. But what does this structure look like in 3-dimensional space? Well, get ready to be amazed, because it's not just a straight ladder, it's a twisted ladder, a double helix. Imagine 2 strands of those nucleotide chains we just built, running alongside each other but in opposite directions. That's what we call anti-parallel. Now picture those strands twisting around each other like a spiral staircase, with those base pairs forming the steps in between. That's the double helix structure of DNA, elegant, efficient, and beautiful. This structure is crucial for DNA's function in storing and transmitting genetic information. But don't just take my word for it, visualizing this structure is key to understanding it. That's where resources like Khan Academy, Crash Course, and Vedantu come in handy. Their animations bring the double helix to life, showing how the base pairs fit snugly inside and how the whole structure twists and turns. Alright, now that we've explored the theoretical side, let's get practical. Remember building things with Legos? Well, it turns out we can do something similar with DNA using molecular modeling kits or online interactive tools. Tools like FET interactive simulations from the University of Colorado let you drag and drop virtual nucleotides, building your own DNA strand piece by piece. You can see firsthand how the sugar-phosphate backbone connects and how those base pairs fit together like puzzle pieces. Trust me, there's nothing quite like building a DNA molecule yourself to really cement the concept in your mind. Plus, it's just plain fun. These interactive exercises take the abstract and make it tangible, allowing you to explore the structure of DNA in a dynamic and engaging way. Okay, time for a little pop quiz. Don't worry, no grades here, just a chance to see how much you've learned. Multiple choice questions and short answer questions are your best friends when it comes to solidifying those key concepts. Look for exercises that challenge your understanding of base pairing rules. For example, if one strand of DNA has the sequence AGCT, what would the complementary strand be? Or, how does the structure of DNA allow it to replicate itself? These types of questions force you to recall and apply the information you've learned. Remember, the goal is not to memorize facts, but to understand the underlying principles. So, don't be afraid to get those brain juices flowing and tackle those practice questions. The more you practice, the more confident you'll become in your understanding of polynucleotide chains. We've spent a good chunk of time diving deep into the structure of polynucleotide chains, but here's the thing: structure is nothing without function. So let's shift gears and explore how the unique architecture of DNA and RNA enables them to carry out their vital roles in the cell. DNA, with its stable double helix structure, is like a secure library of genetic information. The specific sequence of base pairs along the DNA molecule acts as a code, dictating the instructions for building and maintaining an organism. RNA, on the other hand, is more like a messenger, carrying those genetic instructions from the DNA blueprint to the ribosomes, where proteins are synthesized. Understanding how the structure of polynucleotide chains facilitates processes like DNA replication, transcription, DNA to RNA, and translation, RNA to protein, is key to unraveling the complexities of molecular biology. And that, my friends, is a wrap on our journey into the world of polynucleotide chains. We started with the basic building blocks of nucleotides, explored how they link together, marveled at the elegance of the double helix, and even got our hands dirty, virtually, building our own DNA molecules. By approaching the topic from both a theoretical and practical standpoint, you'll be able to internalize the information more effectively. Remember, understanding the structure of polynucleotide chains is fundamental to grasping how genetic information is stored, replicated, and transmitted. It's the foundation upon which much of biology is built. Thanks for watching, and happy studying.